This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 51, the other side of 50. It's September 25th, 2009. Hey, everybody, Vincent Racaniello, and I'm back with Dick Despommier.
Hey, Vince.
And Alan Dove.
Back already.
We missed Alan.
Yeah, been a while.
You missed our golden anniversary.
Yes,
Indeed.
that was a big episode.
So did you, Dick.
I did, actually.
You guys left me high and dry. That's all right. We'll make the what is 75? Sorry.
That is platinum,
Platinum?
I think. Yeah. 50th is gold.
We're going to make 100. It's no problem.
Of course
Just
we
another
will.
year.
Of course. Absolutely.
Bigger and better virology for the next year. You've been well, Alan?
Yeah, things are going well. I actually one of the episodes I missed. I think it was because I was partly because I was just buried in work, which is for a freelancer always a good thing.
Always a good thing.
Great. One of these days you should tell us where your articles get published so we could read them.
Yes. Well, I discovered a number of years ago, the smaller the audience, the bigger the paycheck. So
Nice.
yeah, so I write for trade journals mostly these days. I do some stuff for Nature Medicine.
Cool.
Just did a piece for Nature Methods.
Mhm.
Oh, good.
And then I work for a couple of other trade journals. Drug Discovery and Development is one of my major clients.
We wouldn't be reading those.
No, most likely they're not on your desk.
And probably our listeners wouldn't be reading them.
Probably
either.
not. Well, a few might. If they're in the drug development industry, they probably get it.
Well, that's good to know. The smaller the audience, the bigger the check. That's not the same for TV and the movies, though, right?
No, probably not.
All right. Well, it's good to have you all back again
Yep.
for number 51.
Full
Close
force.
to we got a couple of congratulatory emails, which won't show up for a few weeks because we're so far behind, unfortunately.
Oh, well.
But we are catching up. It's getting better,
Oh,
which
good.
means that we're probably getting fewer emails.
Yeah.
Today, I think we ought to discuss the HIV vaccine
I do too.
trial in Thailand. It may take up the whole time, which would be fine.
It could.
Let's
It's
see,
all over the news, that's for sure.
It's all over the news, and people want to know what's going on. And frankly, it's good that it's in the news, but there are some caveats.
Well, yeah.
There always are, right?
In fact, that's all I was reading about in the news.
So let's see. We got a bunch of alerts. This vaccine, one of the components is a Canary Pox Vector.
Right.
So this is a 2-component vaccine. First, they get a shot of, these are people in Thailand, 16,000 individuals. They get a Canary Pox Vector. And a Canary Pox is a pox virus of birds that doesn't replicate in humans. And actually, earlier today, I chatted with Rich Condit and recorded it, and I'm going to play it.
Great.
In a bit so that people can know what those vectors are. And he tells us the scoop on that. So this Canary Pox Vector, it's an infectious virus vector.
Is this the only time it's ever been used in humans, or are
It's
there
been
other...
used in trials,
It has.
but it's not licensed in humans.
Right.
But of course, they're used in animals for rabies
course.
immunization.
Of course.
And the good thing about this is it doesn't replicate, but it expresses proteins. And in this case, they put in a couple of HIV proteins. They have GP120, which is the surface glycoprotein of a subtype type E, which is prevalent in Thailand.
That's right.
Not in the U.S.
That was one of the caveats of the vaccination.
Yes.
That's right.
So every continent has slightly different subtypes, and you have to immunize against the subtype.
Yep.
Right?
Yep.
So you could view the GP120 as sort of like the HA of influenza. We've talked a lot about that, right? So that's an important protein. But it also has the GP120 is not actually buried in the membrane. It's linked to a second protein called GP41, which is buried in the membrane. Okay, so they also include GP41, in this case from B, subtype B, which is what's prevalent in the U.S.
Oh,
Then they also include a protein called GAG,
right.
GAG, which is the structural proteins of the virion. And finally, the protease. Four proteins.
It's almost the whole virus.
Well, actually, if you express just GAG, you get particles.
Right.
So it's almost, but it doesn't have the nucleic acid, so it would not be infectious.
No.
So they first immunized people with that. So the canary pox would infect your cells and produce all these viral proteins and produce some immunity against them. And then they boost with a totally different second vaccine, which is just a purified GP120, which is made in cell culture, mammalian cell culture. So that's the boost.
So, Vince, when they inject this with the Canary Pox virus vector, is it particle dependent as to how much stuff gets into the cells, or does the canary pox actually replicate?
No, it doesn't replicate.
Okay, so then you can quantify viral particles first and then determine how many cells got infected.
Yes. Whatever you put in is all that gets delivered, right?
And then it dies out.
I mean, the virus gets into the cell.
Right.
It makes proteins. It makes the HIV proteins and a few viral proteins.
Got it.
But then there's no second round.
Right. You don't get any virions produced even.
So it's a one-dose dependent vaccination.
So whatever you put in, it's amplified at the protein level, I would guess. But it doesn't make more. Yeah, it's one cycle. It doesn't make infectious particles. It doesn't spread within you. So separately, these two vaccines had been tried before, and they failed.
Right.
And what they test is, there are two main things that are tested in these trials, whether it prevents infection and whether it makes you less infectious.
Right.
These are very tricky trials, okay? You need a lot of people. You need at-risk people.
Unreliable resource, unfortunately.
And then when you enroll the at-risk people, you have to tell them not to do risky things.
So in Thailand, why is it less risky than here?
Less risky?
Yeah. Who are the people that they're vaccinating?
Okay, so this is a very interesting question. I have a nice little article here just for you.
Uh-huh.
Look, the World Health Organization designated Thailand as a target because it had a huge increase in infection rate, again, in drug users and commercial sex workers.
Now, what do we call those people?
Whores. I'm sorry, prostitutes.
That's correct. Is it legal in this country, Vince?
No.
Yes, it is.
Elliot Spitzer showed it wasn't legal.
It depends on what state you're in.
That's exactly right, Alan.
It's legal in Nevada and Rhode Island.
Rhode Island?
Yes.
I had not known that.
I didn't know that.
That's a little-known fact.
I had not known that.
Yeah.
Well, that's incredible. And do we call them sex workers?
No, we don't, right?
We do not.
Depends on who we are.
We call them residents of the chicken farm.
That's in Nevada. Sure.
So everybody knows that. So
this industry in Thailand,
it's legal.
It's legal, and it made the incidence of the disease increase significantly in the 90s, and that's why they said, let's try it over there.
But you also get a more reliable reporting system because they're not running away from the law.
Right, but also you get infection.
Right.
So here in the U.S., you have a smaller population at risk, and you bring them in and you say, you're going to do this trial. And by the way, you shouldn't engage in risky behavior.
Yeah, right.
Because you're morally obligated, and actually by law, if you're going to do this kind of trial, to tell them that they shouldn't do the behavior that you're looking to prevent the consequences of, right?
Exactly.
But in Thailand, they keep doing it because it's their business.
Correct.
So
Go
ahead.
One effect of that, of course, I mean, obviously you have to do that ethically, but if your educational message has any effect, then these people become less likely to test the vaccine.
That's right. But you do end up getting some people.
Yeah.
I mean, here, I don't remember the number here, but a few years ago, they had 2,000 people enrolled, and there were 100 infections, for example. So that's about the rate that you get.
Right. And in this trial, they got 74 out of the 8,198 placebo recipients became infected.
Right.
51 out of 8,197 vaccinated people. So you do these huge 16,000 plus patient trials, and you get a few dozen people get infected.
And it's a 30% reduction, right?
This is over a period of 3 years, I think, right?
Yeah.
So it's a tough vaccine to try, and you can't repeat this experiment, right?
No.
So
the other thing I'm curious about is, just to interrupt,
sure.
If this is strain specific for Thailand, but most of the customers, now not the sex workers themselves, but most of the customers are from other countries, and they are, I can guarantee that, then why isn't this strain found in other countries then?
It is found in other countries, it's just not prevalent.
So
predominant.
Does that mean that the sex workers are not doing their job right, or
well, they are doing their job right?
But why aren't there more people once the AIDS strain goes to another country, because these people are obviously, I would call them more promiscuous than, let's say, married people, for instance. Why haven't they then spread this virus in their own country? Why hasn't it caught on? Because
Well, they probably have, but against the background of, I mean, taking the U.S. as an example, you have a certain small percentage of the U.S. population will travel to Thailand and pick up possibly HIV clade E and bring it back home, and then they may infect some number of contacts here, but that's a very tiny proportion of the overall population compared to the number of people who will pick it up from other sources within the United States, and it'll be clade B.
Right. I mean, what it does, it tells you a lot about human sexuality.
Does it?
Yeah, by looking at how these things spread among populations. It tells you whether or not this population is contained, or whether it's, as the original AIDS virus was supposed to have been spread from a male flight attendant living in Montreal to whole bunches of other people. I mean, that's what got this strain started to begin with, I guess. So here we have a case where there's a localized population in one country where people travel to that country for that particular activity, and it has not spread that strain to other countries to that great an extent. I find that fascinating.
I'm showing Dick a map of global distribution of subtypes. So here in pink is the Thai. Southeast Asia, right?
Yep.
Pretty restricted, nowhere else. North America.
I would expect to see some, at least in Japan, but that
see
doesn't
it here.
happen either. That's very interesting. It's fascinating.
So those numbers are that's where the press is calling it a 30% protection, which is the difference between the 50 and the 70 individuals.
It's statistically different, but it isn't significant.
So I was talking to my colleague Lynn Enquist, a virologist. He says it's statistically significant, but it means nothing in terms of protection or efficacy of the vaccine. You could never sell this vaccine, and I think no one is trying to. It's just nothing has ever worked at all.
Exactly. It reminds me of the malaria vaccine story, which has a similar poor history.
Yeah, and in fact, I put a little post on my blog when this first came out. I said, this news just puts my jaw on the floor. I mean, it's a result that for many other diseases we would say, oh, that's ridiculous. You wouldn't even publish that.
Right.
But with HIV, because of the litany of failures that have plagued this field, to have any kind of positive result, and this is a positive result. I mean, we're going to spend much of the show dissing it, but
I wouldn't diss it necessarily, but I would want to know, are there biological lessons to be learned from this that you could build on to make it a better vaccine?
Exactly. The real question now is what produced this result out of a trial that, frankly, a lot of people, and I followed the debate when this was first being discussed a few years ago when they were setting up this trial. And I was pretty skeptical of it. It seemed like kind of a silly thing to do. You're going to enroll all these people and you're going to take two vaccines that don't work. And you're going to hope that they work better together. It just, it seemed pretty sketchy. But then you get a result like this, and that suggests that maybe there's some new science we can get out of this.
Exactly.
Yeah, this was a tough trial to sell. We have an article that we'll link to where they had to justify doing this because, as Alan said, both of them had failed.
I'm sure the government structure has something to do with it. The willingness of the sex workers to cooperate have something to do with it. The fact that USAID is concerned and probably supported some of this, although that's just a guess on my part, I don't know for sure. But the best place to try to make an effect on this, though, would be where?
What do you
mean? If you had to pick any one country in the world and concentrate on that one country for a vaccine.
With Africa?
Yeah, that's right. South Africa. You'd certainly pick South Africa.
So, we don't want to diss it, but we do want to make it clear to everyone why it is not the end yet, right? Why a lot more work has to be done. It might be just the beginning. So what is the problem with, why is HIV so difficult? All our viral vaccines work because they generate antibodies and antibody memory, right?
That's right.
And you get infected, you make a memory response, and the antibodies neutralize the virus.
Yep.
So why can't, that's been already tried for years with HIV. They've made GP120 years ago, which is, the HA equivalent. You immunize people, you get antibodies, but they don't neutralize the viruses that are infecting people.
Most of the antibodies that people develop are not protective.
Yeah, they
Right.
don't protect you.
So in the people that have this interesting history of being exposed again and again and again to HIV infected people and yet didn't catch it, like being married to somebody who had it and they didn't catch it, the biology of that hasn't been worked out yet then. Is that true?
Well, there are some people who don't have receptor, remember. That's one part of it, not the whole thing. But then there are some people who do make neutralizing antibodies against other strains. And in fact, there was a couple of papers in August about those, and those may provide some leads.
So do their sera supply temporary protection to people that have high viremias? Can you knock it down by passively transferring that?
I don't believe that's been tried. But the sera has virus in it, right?
No, well, you can remove the virus.
I don't know. I'm not familiar with that enough.
No, but you're going to give it to somebody with a high viremia anyway, so it wouldn't matter. But
I still don't think you can do that.
that's how they proved immunity to malaria to begin with.
Oh, yeah?
That's exactly how they did it.
Yeah. Well, I'm not sure. I would guess no, but
Okay.
I'm guessing.
All right.
So one of the problems is that, these antibodies don't neutralize. And then late in infection, you do get some antibodies again that neutralize the strain that is mainly infecting you, but then the virus changes and it escapes the neutralization.
Exactly.
So there are all these issues.
Yep.
So after these GP120 vaccines failed, they said, well, maybe we need T-cell immunity, cellular immunity. Let's make some CD8, which are the cytotoxic cells that lyse virus-infected cells. And that's the idea of one of these components. In fact, the GP120, this combination of the Canary Pox and the GP120 ends up inducing both T cells and antibodies. And that may be it. The combination may do it. But now it's going to be very hard to figure out what is it about this, because the numbers are so small.
Yes.
And you would like to get those people who were protected and see the kinds of antibodies that they have.
That's right.
I don't know if they can do that.
But another thing that they found in this trial is that the people who got the vaccine but still got the virus went on to develop similar viral titers to the ones who hadn't gotten the vaccine. So it's either protective over some period of time for this 30% that managed to not get the virus, or if you do get the virus, then it doesn't really do much of anything for you. So it doesn't seem to have an effect once you get the virus.
So the people who, when they say 50 got AIDS and 70, this is clinical AIDS, CD4 count defined AIDS, right?
Right.
So what you're saying is
they got it as bad as the people who hadn't been vaccinated.
The same viral titers.
If the vaccine fails, it fails completely, apparently. I mean, but again, this is working with very small numbers. But
so weren't these people followed before they actually got to that level so that you could intervene with the triple therapy?
I do believe that once you're diagnosed in these trials, you have to be given therapy. Yeah, that's one of the things. Yeah. So as soon as they develop it clinically, they take periodic blood and look at CD4 counts, and as soon as there's an indication, I would think that as soon as they see virus infection, they probably would hit them with antivirals.
Yeah, and that's before the CD.
Now, Alan, you said the viral titers were
similar. Yeah, now they saw similar viral titers. I haven't dug deeply enough into the results yet to see when they checked that versus when they're checking the viral titers, they're looking at people who've seroconverted or who are HIV positive.
Yeah,
they haven't necessarily gone to full-blown AIDS.
Right. Probably they wouldn't necessarily, because they'll treat them before they do that.
Sure, right.
Yeah. So maybe the CD4 count isn't the way. They're just looking at virus in the blood. Yeah.
And are they also looking at viral titers against just those two proteins?
No, they're looking at viral RNA by PCR most likely in the blood. Yeah.
Because then you could tell whether there was an anamnestic response or not to those two virus proteins.
Yeah.
Right?
No, they don't. They just look at RNA, and then when they get to a certain level, or when they detect it, they say, "Oh, this person's infected."
And it is possible that the vaccinated people who contracted the virus would have a lesser disease, but of course you can't do that experiment.
That's right.
No.
As soon as they get positive for RNA, you have to treat them.
All right. Yeah. So assume now that these are not people; these are animals that you're trying this out in. And what would you have done differently in the animals than you didn't do in the people to get a more complete picture of what's going on?
Well, first of all, you would inject them with the virus.
No. I mean, you did everything the same as you did here, but what would you do differently that you didn't do with the people?
Well, you could let them go to full-blown AIDS and see if there's a difference,
That's
right?
number one. And number two, you could take, if you wanted, daily or weekly samples
Of course.
of the animal's blood to make sure that the viral titers and the antibodies against the vaccine components were different in the vaccinated than the non-vaccinated group.
Right. Right. You could do a full immunological profile.
Exactly. And none of that was done here. And that's what the, even if it works, if it's strain specific and it just is only protecting people in Thailand, that's not good enough to know that why the vaccine worked. You have to know all the details, right?
Yes.
Sure.
So now you need to know. Now you need to do another trial.
Well,
I think what you do all of these things.
I think what you need to do now is go to an animal model where you can partially replicate this, because we do need some more answers about what's going on at the immunological level in these people.
Absolutely.
I think it's interesting what Tony Fauci said. It's conceivable that we haven't even identified yet what really provides immunity.
Right.
And I think that's true.
Since 1982 to today, that's incredible.
Yeah, we don't know, and now we've got a result where something appears to be working, and we really don't know why.
Well, wait a minute. Don't we have a similar situation with yellow fever vaccine?
Doesn't it make antibodies, though? Don't we understand that?
Well,
yeah, I think we do, Dick.
Yeah, I hope you're right.
I mean, for all the vaccines that work, we know they make antibodies, and that's what protects us. And even for flu, it protects you for a year until the virus changes. But this virus, HIV, changes in you as over the course of years. That's the problem. It's not an acute infection. It's a long-term chronic infection. So there's lots of time for change, and at the same time, the virus is replicating in the cells that you need to make an immune response, right?
Sure.
So that's the problem.
And so in this case, I mean, it might not be the antibody response at all, but the other arm of the immune system, the CD8 response, may be determining whether people get this.
That's right.
So I'm shocked and surprised that we have not taken advantage of the reverse transcriptase enzyme.
Well, we have.
Yeah.
Well, many of the antivirals hit the reverse
No, I mean to
transcriptase.
immunize against that.
Why would you do that, Dick?
Let's see. Why would I do that?
Although, in this Canary Pox vaccine...
Because it's inside the cell and the antibodies won't get inside. Is that what you're saying?
Well, but in this vaccine, included is GAG, the structural genes, and protease. So these are going to be expressed inside the cells.
Right.
But couldn't they be presented on the surface by the
could.
antigen presentation pathway?
So,
and again, the idea is to try to induce the cellular response.
Right.
Well,
against those, I gather.
Okay,
Yes.
so why not include into that then the reverse transcriptase?
You could. In fact, I think there was another Canary Pox I found in my searches that had reverse transcriptase in it.
Yep.
But the thing is, they have 4 proteins, and it's not clear which one is important.
Of course.
And, it's very hard to parse that out in a human trial.
Yeah.
You need to do that first in animals. We don't have a good model.
Well, that's what I was saying.
We have simian immunodeficiency, which is different.
Right.
So it's a real problem.
Yep.
So that's why everybody's very happy about this, right? Because there's some glimmer,
hope.
but
It's the first success, yeah.
it means there's something they did that's correct, but we don't know what.
Right.
I've reviewed grants at NIH like that.
Sure.
Where did they go right?
Yeah, that's right.
It's a line from the producers, by the way.
So there's a very good review article by Tony Fauci from August a year ago, an HIV vaccine challenges and prospects. And he goes through all the things that have been done. It's a nice history. And says a successful vaccine will probably need to induce both antibodies and cytotoxic T lymphocytes. So the CD8 cells that we're talking about. But he says this Thai trial results are expected at the end of 2009. So here it is. We have it. But it's still an interesting article nonetheless.
You can watch the McNeil-Lehrer report tonight, and Tony will obviously be on there being interviewed. And he'll be asked for his opinions as to what he thinks is going on. I'll be very curious to hear what he has to say.
And then how the press interprets what he has to say.
Exactly.
And then I like this here. He says, we may not be able to develop an HIV vaccine that is highly effective in the classic sense of successful viral vaccines. So, we are basing our predictions on what we've done, but this may be very different.
Well, and that also raises a separate challenge, which is if you got a vaccine that worked better than this, how much better would it have to work in order to have a significant impact on the pandemic? People have done some mathematical modeling studies on that. And the ones that I've seen have been pretty grim.
Yes.
If you've got a vaccine that's even 60, 70% effective, it may not really do anything because the disease is spreading so fast already.
Sure. It's interesting, though. You can make two categories out of infectious diseases, all of them. You've got those that vary and those that don't. And when you put those that vary on one side, and HIV is one of them, malaria is another, and African trypanosomiasis is another, and Brucella abortus recurrentis is another. Those organisms are all very difficult to vaccinate against because they've all evolved separate escape mechanisms that have a similar basis. So that's a take-home lesson for us.
Oh, isn't it true that the easy ones always go first, right?
The low-hanging fruit,
low-hanging
as they say.
Yes.
The low-hanging microbes.
This is very interesting because we take the focus away from H1N1 a while, where we have a great vaccine that nobody wants to take, right?
Right.
Or very few, or people are nonchalant. They say, I don't want to take it.
Yep.
And people who have AIDS would love to have such a vaccine.
Yeah, but the virulence factors are quite different here. I think that's the reason.
They're quite different. I understand, but the principle is the same. You can prevent an infection that can kill people.
Absolutely.
There's no point. In the U.S., the 30 to 40,000 deaths a year could almost all be prevented if everyone were immunized.
That's right.
But can't do it, right?
Yeah.
Okay.
So that's interesting. You should mention influenza now because that's the intermediate organism between those that don't vary and those that do. This one varies, but it varies slower. It allows us a chance to catch up to it.
The HIV?
Very, very slow, but we don't even know what it's varying until it's too late.
It's quite slow, yeah.
Malaria is not slow, and neither is trypanosomiasis. So fascinating. On the spectrum of infectious diseases, we cover the waterfront, folks. And all the backwaters,
I think we've covered that sufficiently.
Yeah.
I will play now a clip from Rich Condit to tell us about Canary Pox Vectors.
Hi, Vince.
Hey, Rich, how are you?
I'm good. How are you?
Not too bad. Having a good day down there?
Having a wonderful day.
I'm going to just keep you here for a bit to ask you about Canary Pox Vectors.
Okay.
So maybe you could tell us what is this Canary Pox vector, and is it going to turn us into a canary if we get it?
I don't think so. I think you're going to be okay. Where do you want to start? We can go all the way back to just a bit of pox viruses in general and where this idea of vectoring stuff with pox viruses came from, and work our way to Canary Pox.
Sounds great.
Okay, so let's start at the very beginning and remind folks that pox viruses are large double-stranded DNA containing viruses. They're enveloped. They're very complex, big genome, 200 genes, replicate in the cell cytoplasm. That's unusual for a DNA virus, and it means that they have to encode all of their own transcription and DNA replication machinery. And that's at least marginally relevant for this because it means that all of the transcriptional controls in the virus are self-contained. Okay. So the most notorious of the pox viruses was smallpox, killed a whole bunch of people throughout human history, and is the only disease to have been eradicated. And what was used to eradicate it was a virus, a related virus called vaccinia, originally isolated from nature. It is a pox virus that is closely enough related to smallpox so that if you're immune to vaccinia, you're also immune to smallpox, but distantly enough related so that it causes only a very mild disease. Specifically, if you scratch some into your skin, scratch live vaccinia virus into your skin, usually on your upper arm, you get a single blistering lesion that looks like a solitary version of what covers your whole body if you have smallpox. But under most circumstances, it never travels outside of that lesion, and that heals and you're immune. So that was used to worldwide, ultimately, to eradicate the disease. And smallpox vaccination was discontinued in about 1979. So anybody who's over 40 or certainly over 50 has experienced this, probably has a scar left on their arm, may remember it. I know I had to be vaccinated in order to be admitted to kindergarten, and I remember the vaccination well. In the late 70s, early 80s, several different groups, but most notably a group headed by Enzo Paoletti and sort of co-led by Dennis Panicali, figured out that you could splice genes into vaccinia virus, the vaccine pox virus, under transcriptional regulatory control of the virus, and those foreign genes would be expressed during the vaccinia virus infection.
Now, why would they want to do that?
Well, that was my question. I just missed this completely, okay? Because this is in the early days of gene therapy, and I thought, this is nuts, okay? Because if you want to do gene therapy, you want long-term expression of a gene or something like this. This is a lytic virus. You're going to get transient expression. You're probably not going to get that much made. Who cares? But it turns out, I really missed the boat on this, that you can use these recombinant viruses then to vaccinate an animal for the foreign antigen. And this has been done with tons of different antigens. You can splice in flu, rabies, hepatitis B, whatever. Vaccinate an animal to that, and they're immune not only to smallpox, but that's incidental, they're immune to whatever organism encodes the foreign antigen that you've spliced into them. And this, it works great.
Makes sense because the virus is also already used as a human vaccine.
Absolutely. So especially at the time, it was very attractive because the database on vaccinia is huge. Everybody in the world has been vaccinated. We know what the sort of complication statistics are and what the complications are and how to deal with it. Okay, so it makes perfect sense as a vaccine vector. However, there are some complications. In a certain percentage of cases, you can get a disseminated disease, you can get encephalitis. In a healthy population, about 1 in a million vaccinees will die. That's one of the reasons when the whole biodefense thing was going on, they decided not to do a mass vaccination campaign in the U.S., one of the reasons, because if you do that with 300 million people, you can predict that about 300 people will die. Is that worth it? Under the current circumstances, the answer is no.
Right,
okay,
right.
So there's motivation to make safer vectors. By the way, the simple act of cloning something into vaccinia, depending on where you do it, attenuates it to some extent. The most common site is thymidine kinase, which is a gene that augments nucleotide metabolism, makes available more nucleotides during replication. And in cell culture, that gene's not essential, but in an animal, it does help the infection. So if you knock that out by inserting your foreign gene into it, the virus is attenuated to some extent. But
And
still,
by, by attenuated, you mean it has lowered pathogenicity.
Correct. So efforts have been made over the years to do further research on these vectors and come up with safer versions. And a lot of these were by the same group. Now, a lot of people have done this, but it was really pioneered by Paoletti and his colleagues. They made one derivative of vaccinia that was called NIVAC, where they knocked out a huge number of genes. Okay. They also investigated other pox viruses, notably avian pox viruses, and one of those is canary pox. So canary pox is a pox. Pox viruses are very widely distributed in nature. Most of them are fairly species specific. Some of them cross species to a certain extent. Vaccinia happens to be one that has a very fairly broad host range. At any rate, canary pox is pretty species specific. I don't know how species specific it is within birds, but it doesn't cross out of birds very well.
So what was the logic in looking at other pox species, other animals that have different poxes, that it would be species specific and not affect people?
Yeah. So the idea would be if you could isolate a a pox virus from another species and then stick it into a mammalian species, maybe it would do something, but not much.
Okay.
So that would be.
Did they look at other than bird viruses, or was this the only one?
I don't know.
Okay.
You don't have too many choices.
Yeah.
Well, there's a bunch of, well, I guess birds are vertebrates too. There's a bunch of vertebrate pox viruses, camels, goats, cows, etc. I don't know to what extent these others have been screened.
Okay.
So at any rate, the avian pox viruses, and in particular, we can talk about canary pox, do not productively affect mammalian cells in culture or in the animal. So they do an abortive infection. The virus goes in, gets partway through its life cycle, and for reasons that certainly I don't know, somebody may know, the infection aborts. Some sort of intracellular molecular mismatch. However, it doesn't abort before expressing a significant quantity of, I would presume, early gene products. So that means you can still clone a foreign gene into this thing and under poxvirus transcriptional regulatory elements and infect cells with that and get the foreign gene expressed under circumstances where you're going to get ultimately no infectious virus out the other end. So that makes it a very safe vector.
So when you say express a viral glycoprotein in a canary pox vector, that'll be displayed on the surface of the cell.
Bingo. That's
and
important.
It'll be detected as foreign by the immune system.
Right. And one of the important things about this trial, I think, is that they're using both what is effectively a live vector, that's the canary pox vector, that expresses the protein in the context of an infection, and then boosting with a subunit antigen. So the idea is you're stimulating both arms of the immune system.
Right. So then they also in these canary pox vectors, they have protease and structural proteins of the virus, which normally would not be displayed on the membrane as a glycoprotein.
Right.
But I assume that these are going to, peptides derived from these proteins will be displayed on the cell.
Right. I would assume so as well. Yes.
And they would contribute to the immune response.
Right.
So these canary pox vectors, are any used in vaccines, or are they all at the testing stage right now?
In fact, they are used pretty widely as veterinary vaccines. Last time I took my cat down to get her vaccinated for rabies, I asked about the vaccine, and they said, sure enough, it was a canary pox vector expressing a rabies glycoprotein G.
So we know that these vectors give sufficient immune stimulus to get good protection right, against the virus like rabies. And so this could be the first one licensed for humans, though, right?
Right. I have not seen, other than these trials, pox viruses being used in humans, and you can kind of understand why.
Sure.
It brings up all sorts of scary scenarios, but in particular with these attenuated vectors and the Avian Pox vectors, I think they're going to be pretty safe, very safe, and so there's potential to expand their use in humans.
So do you think that the use of the purified protein, the GP120 as the boost here, is in part because you can't go back again with the canary pox vector because you may have developed immunity to it?
This is something that I need to look into more. There's been significant study on this, and it's an important issue. Once you immunize with a pox virus vector, does it compromise the second immunization because you've mounted an immune response to the vector to start with? Here I'm not an expert, okay? And I think the answer is no, you're not totally compromised, but there may be some compromise. I'll just have to research that more, or maybe somebody will call in and give us the answer.
You can imagine that you could not use a canary vector for multiple immunizations. This might be your only shot.
Right. But I think as important in this is the idea of having two different types of immunization, one with a live vector and one with a subunit vaccine, to try and balance the immune response. You get a different kind of immune response in both cases.
Right. Well, in fact, these two vaccines have been used separately in clinical trials and had no effect on their own. So there may be something to that. The effect in this case is very small, but it's something, and maybe it's something to build on.
Right.
Are there other pox viruses that are used as vectors as this one, or is this the sole one?
I think there are some other Avian Pox vectors. I'm not sure about that. I'll have to look into that more. And then there's a variety of different attenuated things that are based on vaccinia to start with. As I said, there's NIVAC that's been deliberately engineered to knock out a slug of essential genes. There's another virus that's going to be probably the, I guess you'd now call it third generation. I would assume it's going to be the third generation smallpox vaccine called MVA, which is a vaccine where they started with vaccinia virus and it passaged it hundreds of times in, I believe, eggs. I don't know if they put cell culture passages in there or not as well. And like many other attenuated vaccines, came up with a virus that was attenuated. It's lost a lot of genes and that kind of stuff. So there are other vectors out there.
So who were the two who originally developed this again? One was Paoletti, right?
Paoletti, Enzo Paoletti, and Dennis Panicali.
Dennis
they worked together.
Yeah. So they were up at in Albany, is that correct?
Yeah, they actually started a company. Enzo started a company called Virogenetics. He's no longer with them, and I think that Virogenetics, this Aventis Pasteur, is another incarnation of Virogenetics, or absorbed them, or something like that. I may not have that information correct, but at any rate, that's where it all started.
No, that's great. We're grateful to them for making this. It's really
absolutely
good. And I think it's important for people to understand the origin of this canary pox, because you can imagine that the press is not going to get it right in the next couple of weeks, and people are going to be wondering, why do I need a bird virus? So,
well, the other thing that occurred to me in thinking about this is this is a wonderful example of how basic research translates into something that's medically relevant down the road. Okay, because you can even ask questions about, is it worthwhile to study transcriptional regulations in pox viruses? We've eradicated the disease. And some people would say no to that. But, you might say yes to that. But the other question is it worthwhile to study a pox virus of canaries, for crying out loud? Who cares about canaries, much less their pox viruses? And here we are. With a very relevant use of all that stuff.
Well, the problem is, as you know, Rich, with limited resources, you have to make tough decisions, right?
Yep, absolutely.
And it's easy to make the easy decisions, right? You can work on human pathogens. That's no brainer. The real tough one is whether you work on a mouse virus or a bird virus, because you never know what it's going to bring down the road.
Right.
And if you said go ahead, then in hindsight, you'd look brilliant. Anyway, I think that's great. We appreciate you joining us at short notice.
Great.
And you go off and have a great weekend, and we'll see you.
Sure will.
We'll see you in TWiV in a couple of weeks.
All right, I'll be looking forward to it.
Thanks, Rich.
Yeah, sure.
Bye-bye.
Bye.
That brings us back to live again. I'm going to talk with Scott Hammer next week. I'm going to his office and recording a half hour on HIV vaccines and clinical trials, because he runs them here. He's agreed to see me for a half hour.
Superb.
Awesome.
And we'll stick that up sometime for everyone to hear. And we'll do just
now that is something that I've noticed. Depending on what I mentioned, that the smaller the audience, the bigger the paycheck. But the disadvantage to working for obscure outlets that people haven't heard of is that they're less likely to return your phone calls. So, I mean, in your case, you're with TWiV, so the door opens for you.
Well, I think it's because he knows me. I don't think he knows what
that. Yeah.
To be honest, guys, I can get doors open because I'm a virologist and people know me, as you know, and Scott knows me, and whoever.
I was just trying to hype the show.
Yeah, I think I would really love if that were the case. Maybe someday. Oh, TWiV, yeah, I'll be on TWiV.
Heck.
But now I'm leveraging my 27
Tell Larry
years.
King to hold.
That's right. Maybe YouTube can open for us someday.
I was actually.
Did I say YouTube or YouTube?
Listen, I was actually YouTube. I was actually going to bring a video camera here today and tape it and put it up on YouTube.
Oh, I'm grateful that you didn't. Vince.
Why? You're such a handsome guy.
Stop it.
Plus, you're a good cook.
All right, I'll take that.
We will do just a couple more stories. But before that, I would like to mention our friends at Citrix, who are sponsors of this episode. And, of course, Citrix is the maker of Go to My PC. Balancing demands of your job and family is very challenging. And we think you can dramatically improve your work flexibility by using Go to My PC to access your work computer from home. Now, you're probably thinking, what is he talking about? How does this have anything to do with my job and family? Well,
yeah, only the people that haven't heard us would say that, actually.
probably everyone else is pausing now and fast forwarding. But if you have all your stuff on your work computer, when you go home, you can't access it. You could go home a little earlier. You could take a day off a week if you could access that material. And Go to My PC lets you do that. It's a very simple program you install on your work computer by going to their website and clicking a few buttons. Then you go home or to any computer with an internet connection. You go to their website, put in your login, and boom, there's your computer. You can run all the programs, get all your files, do email, network resources. Amazing. And this can change your life and your family, and your kids will love you. So you should try this now. It's very easy to do. You can go to gotomypc.com/podcast. That's gotomypc.com/podcast. You get a free 30-day trial, and we thank them for their support. I wanted to revisit fish flu, just briefly. Do you remember fish flu?
I do.
TWiV 41. We talked about the fact that Chilean salmon industry is getting decimated by infectious salmon anemia virus.
Careful how you use the word decimated, Vince. That only means one fish out of 10.
Gosh, I remember that from TWiV 1. Well, I didn't obviously remember it, so that's the problem. It's,
okay,
Alan,
maybe it's being heptimated. We don't exactly know. But
sorry. They're having a problem. So their tonnage went from 400,000 tons a year to 250,000 tons. So that's not decimating. You're right there. But it hurts because they sell a lot of salmon.
Declinating?
Maybe.
Now, these are what strain of salmon are they? Did they say here?
I don't know. We probably mentioned it back in TWiV 41.
Salmon are not native below the equator.
No, these guys do it farming offshore in pens, right?
Yep.
So this virus, infectious salmon anemia, is actually a member of the same family that influenza is a member of. Segmented RNA virus with an envelope.
Right.
Now the wire story on this says that the virus is specific to Atlantic salmon.
Atlantic.
I don't know if that's accurate, but
it might be. I think that's what they're raising here. Is Salmo solar is the actual name of that fish, whereas the Pacific salmon's genus name is Oncorhynchus. So if they were to switch salmon species, they may be able to get rid of it.
Well, I know that Norway also had a problem with...
that's Salmo solar, too.
With it's the same salmon?
Yep.
Okay. Mm-hmm. So that when they farmed them, they used the same salmon all over the globe. Okay. Well, anyway, this virus was a problem we talked about. I just wanted to mention that now they're using a vaccine in Chile. And this is, I believe it's an inactivated vaccine, so it's produced in cells and inactivated, much like the influenza vaccine. But the interesting part here is that they have to inject this into the belly of each fish individually. It's injected into the bellies of young salmon while they are still in their freshwater phase.
Right.
They weigh just a few dozen grams.
They're called par at that point.
So they have a freshwater phase. So they have to keep them in fresh water to a certain size.
That's correct.
And then transfer them to the ocean.
It's just what happens in nature.
But no, for these farms, they have to transfer
No, they
them.
have to imitate what happens in nature.
Oh, they imitate. I'm sorry.
That's right.
So they put them in trucks and they ship them to the ocean.
Yep.
But look, they've done 300,000 hatchlings. There's a picture here of the guy just injecting each one.
Yeah.
I mean, that's not really a practical solution. They hope to inject 10 to 12 million fish in 6 months.
So where do they think this virus came from?
Oh, from finfish in the ocean. Yeah.
Salmon specific?
It's not. It infects many different fish.
Right. Okay. So that's the answer then.
It just happened to come from fish that when they put the salmon in the ocean, it infects
and
them.
I think we previously talked about the issue that these farmed salmon are probably more susceptible to
Of
it
course.
because they're under such great stress.
That's
They've had
right.
the wild bred out of them. That's right.
But so this is a good. If you want work inoculating salmon, there seems that there's a lot of work. Or we could do research on figuring out Maybe throw the vaccine right in the water.
Switch species, for God's sakes.
Not raise salmon.
No, make it Oncorhynchus instead. The Pacific salmon doesn't get this virus.
Well,
Probably
but is it wise to start farming a non-native species in a different ocean?
Well, that's what you've done here.
I mean, not that wisdom has much to do with any of our food policy.
Chile is on the Pacific coast, so if you took a Pacific salmon, the answer would be...
Oh, you're right. You're right.
So therefore, they have taken a different salmon into the different ocean.
Already.
The horse is out of the barn, right?
Well, okay. Of course. That's right.
It sounds to me like a good idea, but there's probably an economic reason why.
Yeah, because Atlantic salmon tastes better.
Okay, there you go. Nobody's going to buy the other one.
Well, they
This is really fatty stuff.
Very
it's got omega-3 fatty acids.
It does.
Which are good.
It does, but there are 4 different kinds of omega fatty acids.
Well, aren't they all good?
They're all good for you, but you have to have them all to be good.
Okay. Yeah, so I just wanted to mention that briefly because they're getting immunized. There is a vaccine.
Oh, you perked my interest.
And
David Baltimore's too. I hope he's listening because he's a fisherman as well.
There has got to be a better way to immunize these fish.
Exactly.
All right, one more story. These are 2 out of the mainstream stories because this one,
no pun
I think
intended.
This was Alan's find. Why don't you do this story, Alan?
Oh yeah, the blueberry virus. I found this a couple of weeks ago, and then I proceeded to miss the episodes until now. So there's a virus stalking the nation's blueberry crop, and this blueberry shock virus. I just love the names of plant viruses. They're so evolved.
Oh yeah, absolutely.
Blueberry shock virus is a single-stranded RNA virus, unenveloped, and it affects blueberry plants. It also affects a few other species, or can affect them. But it appears to be pollen-borne. So this is a
the
problem.
bee is a mechanical transmitter then,
You
eh?
pretty much can't contain it once it hits a field, because if you go around eliminating the plants that have symptoms, you're already too late. The bees have spread the pollen all over the field, and all the plants have it.
Interesting.
And this has now, the story that brought it across my radar was it has now hit Michigan. Previously, it was only on the West Coast. They had found it in Oregon, a couple of other places. And it turns out Michigan is the #1 producer of blueberries.
Fascinating.
So
high bush or low bush blueberry?
I do not know whether they do the northern high bush or the northern low bush. I know southern high bush has become quite popular in warmer climes, but
Is up by where you live? It's a low bush industry, isn't it?
Northern, I think Massachusetts is mostly northern low bush.
Yep, in Maine, of course.
Right.
So what does it actually do to the
I was going to ask that too.
Well, it's interesting. It doesn't kill the plant.
It shocks them.
It.
With what?
So it causes the standard shock response in these plants, which occurs right during the blossom season because it's pollen-borne. The affected plant opens its flowers and then suddenly drops them.
Really?
And in a lot of cases, it looks like they get red spots on the leaves, and some other things happen that clearly indicate that the plant is going into this shock response.
Got it.
But then the plant lives. It just has very low yields that year.
Yep.
And for the ensuing year or two, it also has low yields, and then it recovers.
Right.
But it remains a reservoir. So it continues to infect surrounding plants, and if you plant new plants around it, of course, they'll get infected, which is why once it hits a field, you're pretty much done. It's going to spread to all the plants once you've seen symptoms in any of them. It's too late.
Fascinating.
And all the ones that have been infected will recover, but then they will be carriers, and so it'll become. It seems like it's a virus that's in the process of becoming endemic.
So you have to destroy the crop?
I guess you could either, harking back to the previous story, you could switch to a different strain. I don't know if I don't know if any of the other strains of blueberry are tolerant.
I hate to say this, but it's a consequence of monoculture.
Please explain.
Well, the moment you raise a lot of one thing in a small space, it rapidly becomes discovered by all of the things that want access to it.
Like swine.
Yeah, exactly like that, or like us as
Although,
humans.
In the case of a pollen-borne virus, I would expect that would affect it no matter what, because the plants have to be able to spread pollen between themselves.
Yeah, this is an absolute requirement for this plant to fruit.
Yeah.
That it must be pollinated.
So I'm sure monoculture is not helping the matter, but that may not be.
Well, there's a real danger of this one. I wanted to just point this out. It sounds peripheral to the story, but it's actually not. There's a plant like blueberries that grows up through Canada called the Saskatoon. I don't know if you're familiar with it. It tastes like a blueberry. It looks like a blueberry, but it grows on high bushes. And it's related to blueberry. I'm convinced of that because of its taste and its size and its shape and everything else suggests that when there are no Saskatoons, the grizzly bears of the area seek other food. One year they had a killing...
Oh dear.
Yeah, exactly. They had a killing frost in June once, which killed off all the flowers for the Saskatoons that year. And the following fall of the same year, 4 fishermen who had nothing more in common with the environment than other than to be there were attacked and killed and cached by grizzly bears that were so hungry for anything that they started to seek out human prey.
Now, I didn't know you could cache a dead fisherman. I thought it was only Czechs.
Well, there are some banks. There are some river banks that will actually cache that, Alan.
Wow.
So it was a real fearful deal around September, October, early November to be on a river trying to catch a trout and be worried about whether a grizzly bear was actually stalking you.
Well, now you could just combine this into a 2-sport trip by packing a rifle, right?
Yeah. What's the rifle for? To shoot yourself when the grizzly bear charges you.
No, but they don't charge you. They rely on cash.
Stop.
So how do we deal with this blueberry virus? So
Right.
you could kill all the crops, you could substitute. Maybe you could make resistant crops. I'm not sure that that's possible. Or you could make antivirals. But
Yeah.
I don't think people do that typically, right?
I'm
Right.
not aware of any antivirals for plants. They'd be
No.
pretty complex to administer.
Yep. So resistance is the key.
Actually,
So
if you had an antiviral and it could be included into a pollen grain,
Yeah.
you could get the bees to do it for you.
So we have some listeners who work on plant viruses. For something like this, tell us what you would do to get around it. And send us an audio file. That would be great. We could play it.
And switching to strawberries is not an option, by the way.
Right.
No, no.
I like blueberries. I have to correct you, Alan. I'm really sorry. This is it's non-enveloped. Did you say it was enveloped?
I said it was
All right.
non-enveloped.
Non-enveloped. It's an RNA virus.
It's a naked icosahedral
Yeah.
RNA virus.
It's actually plus-stranded RNA. For those of you who remember our polarity
There are
lectures.
7 different kinds altogether.
Yes. Okay, let's move to some great email. We have an email from Didier. Here are some papers related to different subjects you talk about in your recent podcast. The first one is an interesting paper on how to calculate the cost of the development of a vaccine. I think we talked about that. In this case, rotavirus vaccine, and the difference between the development cost of a drug and a vaccine. I discovered in this paper the very interesting notion of cost of capital and the observation that the most expensive part of vaccine development is at the final steps, phase 3, when the risk to fail is lower. The risk to fail is lower. I think it would be higher at the end. You've put so much money into it, right?
But you already know it's going to succeed.
The risk to fail by the time you get to phase 3.
Okay,
Yeah,
got
you
it.
should know it
Not
works.
the cost of failing, the risk.
Yeah.
All right.
Yeah.
Right, the cost of failing is much
Got it.
higher.
The cost of capital is not a real out-of-pocket expense like interest on a loan, but rather the estimated profits foregone by using funds for drug development rather than simply investing them based on a notional 11% compound rate of return.
It's the opportunity cost of spending.
That's right. That's absolutely right. Also, with rotavirus, there's a very interesting paper on the seasonality of infection. In that case, the key is not temperature or humidity as for flu, but a spatiotemporal variation in the birth rate.
Right.
So he sent us a link to a paper in Science on this, that they did some modeling. This is a modeling paper. They modeled these rotavirus epidemics, which occur in very specific times. Rotavirus activity in the U.S. starts in the Southwest in late fall and ends in the Northeast 3 months later.
Fast.
And they found it correlates with the birth rate, basically.
Very interesting.
So we'll link to that because I can't explain it. The epidemiology and mathematical modeling. I'm a virologist.
Hey.
And then he talks a little bit about virus receptors and sugar. The calicivirus, and particularly the TWiV famous Norwalk virus. You got
It's now famous.
TWiV famous is interesting. Your susceptibility to one strain of Norwalk depends on your blood sugars. or blood groups. Don't worry, there is a Norwalk virus strain for each blood group.
Sounds
In fact,
like malaria.
Some people think it might be a receptor, but not clear.
Which blood groups are protective?
No, it depends on the serotype.
So you have to line them all up and then go through the
They
blood
all
groups.
get different strains infect different blood groups.
Because there are strains of malaria, or species of malaria, which are blood group specific, tolerant.
And then he made a statement which he says, I was involved in this work, so don't cite it, so I can't read it. Thanks for your podcast and your blog. Long life to TWiV. Oh, that's good. Long life.
Great.
Peter wrote, Vincent, Dick et al. I discovered TWiV over a week ago and must have listened to 20 issues already. I'm an avid listener of Futures in Biotech, too. I've been interested in molecular biology for quite a while now. Well, I guess it's in the widest possible definition. I've read Eric Kandel's book and was in awe about the molecular workings of a neuron. I've read Dawkins' extended phenotype, and the fine balance of coevolution between parasites and their hosts struck me as amazingly beautiful. And now your work on viruses and how they interact with the cellular machinery, it's all just so, well, I guess I have no better word than beautiful. Life has evolved over quite a bit of time and taken many different routes. From where I'm now, I'm willing to agree with Dawkins that life is really just self-replicating information in the form of nucleic acids that has created survival machines of various grades of complexity. Anybody read Dawkins?
I haven't read him, but I know his philosophy.
It's a nice statement. From an engineering standpoint, viruses seem to be the most elegant of those machines, since they use the least effort and still, or maybe for that exact reason, are tremendously successful. Well, their effort is done by the cell, right? The cell takes care of everything. The virus is just an instruction sheet. Do we have any theory on where viruses stand with relation to the tree of evolution? Obviously, we know little to nothing about the early days of DNA-RNA-based life, but is it possible that cells and viruses have a common ancestor, or do viruses have a completely separate root from what we call life?
The answer is yes.
Yeah. So we actually talked about this with Luis Villarreal on an older TWiV, so check that out. I have a blog post on viruses and the tree of life, which talks about this. Viruses have many origins. They're polyphyletic, which is different from all other stuff. I'm not going to say life because they're not life, but so it's hard to put them on a tree. But there are many reasons why you can't, and I want to refer you to that blog post.
Yeah.
Thanks for the great show. As you can probably tell, I'm just a layman standing on the sidelines, taking in as much scientific information as possible, marveling at the beautiful complexity of what we call life.
How wonderful.
It's okay. Not just a layman. You're a person. Right?
Not just a layperson either. You're somebody who's interested in who you are and where you came from and where you might be going.
That's right.
And that's wonderful to hear. Absolutely wonderful.
And we appreciate your feedback. Allison wrote, "Hello, I hope you will forgive any spelling mistakes as I am writing this on my iPhone during my normal train ride commute to Philly. I'm also a bit behind in my TWiV podcast due entirely to the discussion on colony collapse disorder, CCD, on last week's episode, which prompted my renewed interest. Scientific American has done about 2 to 3 recent podcasts about this, which have been really good. They include a couple of interviews with Dr. May Baumbaum on CCD, as well as the beekeeper from Darwin's house. The first interview with Dr. May Baumbaum, she takes a CSI-type approach to explaining the current understandings on what causes CCD. The following are notes I took on my iPhone while listening to the interview. So basically, this is a paper we discussed a few weeks ago in PNAS. Bees have a lot of problems. Israeli acute paralysis virus it was suggested to be a cause of colony collapse, but it's not found in all. It's found in healthy colonies as well. Bees also have various blood-sucking parasites. I thought Dick would like this and might know about it. You know about blood-sucking parasites?
It has aphids that actually, and mites. Mites, I meant to say mites. I didn't mean aphids. I take it back. I meant mites. Bee mites that actually can exsanguinate a bee.
They also found Nosema cerani virus. Sounded like it came from a fungus, new but also found in healthy bees. They looked at the effects of pesticides, new and old. However, they have been used for a long time, and CCD is rather recent.
Right.
And then they have a new way to study this by using the honeybee genome. And that's where this paper came in. They found that the most common thing in all these bees in these CCD colonies is that their ribosomes are disrupted. And so she thinks they're infected with multiple viruses and maybe picornaviruses, and these lead to disruption of ribosomes. Although from my perspective, Allison writes, I don't understand how the ribosome damage causes them to disappear rather than die. It's a good point. I hope this helps and brings up more questions. Personally,
well, they probably do die and then they get eaten, or they slow down and then they get eaten. There are a lot of predators of honeybees.
Yeah, but the thing, that's the assumption. They disappear from the hive, right?
Right.
We don't know what happens to them.
No.
Right. They go out foraging, and they're either sick or lost or what have you, and the preying madness.
I want to say something here because I actually knew one of the students of Karl von Frisch. Karl von Frisch was the man in Germany who worked out the waggle dance for honeybees. And the waggle dance actually tells you the direction and the intensity of the source of honey. And the guy I knew was at the University of Notre Dame, and I'm trying to think of his last name. His last name was Esch. And he gave a demonstration up at the Bronx Botanical Gardens as part of the Zoological Society. And we actually watched the bees come back. He could tag them at various distances and with varying intensities of honey, or in this case, sugar solution. And you could watch the bee that was he put a yellow dot on a bee that was drinking from this concentration of sugar, a red dot for twice the concentration, and a purple dot for 3 times the concentration. And it was a 1-to-1 correlation that the bee would come back to the hive and do this waggle dance, either more intensely or less intensely, depending on the source of the honey. I'll bet you that this disease, whatever they have, it causes them to lose their orientation once they're outside, so they can't find their way back to the hive, and that's going to be the big problem. So
they have to be able to go back and shake their honey maker.
Exactly right. That's the deal. So it's a disorientation that results in the loss of the bees, and then they get lost. It's like the salmon. When they try to come back to rivers that have been polluted, they come back based on the smell of the river. When you take the smell away, of course, the salmon get lost. Eventually, they end up dying in the ocean.
Could have multiple etiologies.
Exactly right.
So here's the issue I have. So the conclusion of this PNAS paper, and you may remember this, Alan. You remember picornavirus was when they infect cells, they shut off protein synthesis. As a consequence, the polyribosomes dissociate. The ribosomes don't dissociate because they have to translate the viral message, but the polysomes that are translating cellular mRNAs come apart. What they find here is that the ribosomal RNA is fragmented. This is not a consequence of picornavirus infection, as far as I know. It's the shutoff is, but not degrading ribosomes. So I'm not sure that this really indicates a picornaviral etiology of this disease.
Right.
But what do I know?
To return to the statements that I was making, I just thought of something else, of course. And that is that if you were interested in knowing whether or not your bee colony was going to exhibit colony collapse disorder before it did, this waggle dance test may have some significance here. That is to say, at some point, the bees will begin to lose their ability to give off signals and to tell the directionality of where the honey comes from.
Right.
So you might be able to work this out as a laboratory test for knowing whether the bees are actually sick to begin with or not by just knowing whether they can find their way back to the hive.
Well, now there's another complication here, though, is that the hives that are experiencing this that I've heard about are the ones that are being maintained commercially.
That's correct.
And there's been some discussion that people who keep a couple of hives in their backyard have not really seen this problem.
Yeah,
which suggests maybe there's something, either the stresses of being trucked around the country or the fact that they're fed a diet of high fructose corn syrup instead of their regular.
Exactly.
I mean, seriously, this is
you're right.
Feed to the bees in a commercial setting, but people who are doing it at home don't do this.
You're right.
And these are all the bees that are being affected are being maintained in very different circumstances from anything resembling their wild setting. So that it may be simply that stress is making them more susceptible to all these conditions.
And they winter over in Texas. So so the point is that what you were raising before about that blueberry shock, I mean, you should probably also check the bees down in Texas to see if they've got this stuff hidden away in some of their pollen to make sure that they can't be transmitters of this thing too, right? I mean,
since they're apparently carrying everything else, yes.
Yeah,
right.
Sure
So eventually everything connects to everything else.
All right. Thanks, Allison. As you see, we got a discussion out of that.
Oh, yeah.
Arson wrote, "Hey, I was just listening to TWiV 47 where you were discussing the prospect of reemergence of viruses. You were arguing that if viruses would suddenly disappear, they would not reemerge." I said that.
You did.
And you asked the question, Dick, I think.
I did.
I was asking myself that question some time ago, and I came to the opposite conclusion.
Uh-huh.
It is my understanding that viruses have emerged from accumulated errors in bacteria's own genetic code. As a programmer, I was playing with evolutionary algorithms for some time. The thing is that although those algorithms are driven by random processes, they can come to exactly the same or very similar solution. Here's an article stating that big brains have evolved twice within our own evolution. It links to an article about this. Viruses have emerged from errors in the bacteria's own genetic code. That's one theory, that phages emerged from bacteria. Sure, it's the same with eukaryotic viruses, but we don't know, in fact. So yes, if that is how they emerged, then that could happen if you eliminated all of them today, right?
And by the way, abstract reasoning has also evolved multiple times.
That's right. It has.
Yep.
I suggest that viruses can and do emerge spontaneously as well, but there is a limited number of optimal configurations in which the viruses can optimally reproduce and infect a lot of hosts. So every new virus which emerges evolves into one of those configurations. Our own cells have fragments of ancient retroviruses embedded into their genetic code. It doesn't seem too far-fetched to me that those fragments sometimes get activated and recombined. So I think, yes, the whole idea that we have only so many different genome configurations. How many is that, Dick?
7.
7. I think we would only see 7 coming out again, no more.
A question then is, if you were to keep a chemostat of E. coli going indefinitely, would you actually be able to watch this?
I don't know. May not have enough time. May not be the right conditions. Could be.
I mean, every 20 minutes.
I don't know. It's a good question.
You keep it in log phase growth for long enough, and you might be able to get something going.
We have endogenous retroviruses, pieces of dead ones, and they could recombine, yeah. So I think our original posit thing was that we got rid of all those too, which would be really hard. So those could give rise to virus. But what would give rise to a picornavirus? Where would that come from? I mean, so we have nothing in cells that look like them right now, so they can't come.
That we know of.
That we know of, yes.
That we know of, that's
Well,
right.
we do have some odd infectious things like prions, where you've got a protein that's capable of modifying another protein to look like it, and we have fragments of RNAs that are catalytic. It seems to me like you could put together something resembling an RNA virus if you had enough time.
Has anyone ever done the proteomics of viral protein, individual viral proteins on the capsids, rather, to see if they matched in at least 20 to 40 to 60 to 80, who knows what, with any of the normal functional proteins that you might find in normal cells?
So not sequence, no, but maybe there are motifs that may be similar. Folds, protein folds for sure are similar, but not sequences, no. So that's the problem. There's nothing we know. But you could imagine, as Alan said, that new things could, but they would be new, not resembling maybe anything that we know of.
Right, but you think about all of the other complex structures that have evolved. I mean, the thing about evolution is that it's had billions of years to work on this, and given that quantity of time,
that's right.
If I were a betting man, I would place my money on viruses reemerging.
And we know so little about the structure of most of life on Earth anyway, so that it's tough to make generalizations at this time.
Yeah, but the thing, I agree, but when viruses emerged at a time, maybe things were very different, and that was a perfect environment for it, a chemical environment, a soup, and they wouldn't emerge. I just don't know.
Have I asked the question yet whether extremophiles have their own set of viruses?
Yeah, they do.
They do. Even the ones coming out of the hot vents?
Oh, yeah. We should actually do a show on those. That would be cool. Yeah,
cool?
they do. Those hot vents, they have hot viruses. Absolutely.
Cool topics from hot vents.
All right. The last email is from Ricardo. I'm a fourth-year medical student in Brazil. I'm an avid listener of TWiV, and I'm currently involved in some clinical infectology research about the swine flu pandemic. I live in a state called Paraná in the south equals cold part of Brazil, where the swine flu is a big deal, with 4,931 confirmed cases, 195 deaths so far. The catch is that our state has only 10 million people, and we basically only had the lab tests done on severe cases or unknown respiratory deaths. Anyways, we had 3 or 4 cases which were very severe, progressing badly despite treatment in the ICU, clinically resembled all the other severe cases of swine flu, but the lab results of RT-PCR came back as seasonal influenza. Repeat testing confirmed once again seasonal influenza. Now we are on to sequencing these samples because we find it hard to believe that seasonal influenza could account for these severe acute respiratory syndromes of all these patients, exactly in the 3 to 4 worst weeks of the outbreak. Normally in our service we have 2 to 3 SARS of unknown origin despite investigation. However, we did not, for monetary reasons, use to search for viruses per year. This time we had these 4 cases just in our hospital in a month. Our question is, could a co-infection of the new strain and the seasonal strain of H1N1, when using the current WHO CDC primers for RT-PCR, be amplified in such a way that only the dominant strain would appear as being positive in that sample? I was told by my teachers that this indeed happens in some HIV patients infected with multiple HIV quasi-species that have different susceptibility to antiretroviral treatments. We'd really like to hear your thoughts on this. Best regards.
So you'd have to do separate PCR reactions for them.
Yeah, I think, well, this is an older email. He's probably sequenced it by now, and that'll tell you the answer.
Yeah.
But I would think you could cross-prime. It depends what primers you use. So I asked Raul here, who is our sequencing guru, bioinformatic guru, and he said that it could happen that you amplify the wrong H1N1, and you have to design primers that are specific, but that can be done. So I looked on the WHO site. They have their primers. They have a kit. You get primers for universal influenza A, you get swine flu A, you get swine H1, and then you get a control. So even with that, you could imagine that you could amplify the wrong one. And you're just looking at a band on a gel. You're not sequencing, so the answer is yes, but you probably already know the answer. You know, it sounds to me like those are not seasonal influenza cases.
I would guess that's right.
But it could be. So, Ricardo, let us know what you found because you've already probably done it by now. All right, that brings us to our science picks of the week.
Yeah.
Alan, what have you got?
I have a book that I think Dick will appreciate. It's a book called Bat Rabies and Other lyssavirus Infections.
Nice.
This was just put out a couple of weeks ago by the U.S. Geological Survey National Wildlife Health Center.
Very cool.
And as a government document, it's available for free. So
Neat.
The price is right, too.
Dick likes it even better now.
Yes, exactly. You can download a free PDF. You can order a paper version of it. You probably have to pay shipping on that. They may charge some kind of fee, but why would you do that? You can download the PDF. And it is it's a surprisingly readable book, or booklet. I guess it's 80 some odd pages or something. So it's not, it's covers a lot of the, covers the science behind it, but it's not a dry review that gets you really bogged down. I mean, it really dives in, and I found it pretty readable.
Lovely.
And really covers the territory quite nicely.
Is Peter Daszak involved in that at all?
He may have been. It's one of these by a cast of thousands, I think.
Right. And seasonally topical also, with Halloween just around the corner.
Yes, it is, absolutely. So the named author is Denny Constantine, who, I don't know, maybe Denny was responsible for this whole thing, in which case, kudos, Denny. You did a nice job. It's quite a book.
Neat stuff.
Yeah.
Cool. That's nice. I like that. Yeah, you had picked that a few weeks ago, but you didn't...
and I didn't show up.
So now that's great. Good. Excellent. Dick, have you got something for us?
So you might say Alan rushed in like a bat out of hell. The answer is yes, I do, actually. This month's issue of Scientific American has a very nice description of the use of adjuvants for stimulating higher degrees of immunity to all kinds of infectious disease vaccines, but particularly viruses. They also mentioned other agents as well. But they concentrated most of their attention on viruses. So if you want a good review of what an adjuvant is and what it does and how it might enhance an already good immune response, I highly recommend that.
Do they have an online version?
They do.
Do you have to subscribe?
You do.
What is this?
What can I tell you? They're a commercial venture.
Actually, you can get the article. I'm looking at it right now on the web.
Okay.
Looks like you can.
I'll take that all back then.
Boosting vaccine power.
Yep.
Hey, sounds great, Dick.
Boosting vaccine power. That's
Sounds
right.
good. I'll
And
read
so
it tonight.
Because we had a TWiV on this, and so I just thought this would be a good follow.
Well, I think adjuvants are a great topic.
Yep.
And this is a substantial article. It's
It is.
Yeah, it's like a 6-page
That's right.
online.
Who's the author?
Natalie Garçon and Michelle Goldman.
Indeed.
Okay. We'll check it out. That's good. Thank you, Dick.
You're welcome.
I have a kind of a tongue-in-cheek recommendation because in the next few weeks, the Nobel Prizes will be announced.
And more importantly,
The Ig Nobel Prizes.
yes.
The Ig Nobel Prizes.
That's right.
So that's the book called The Ig Nobel Prize by Mark Abrams. So it's funny anti-Nobel prizes, would you say?
For inventions gone wrong.
Yes.
The official logo is that it's research that cannot or should not be reproduced.
Right. Remember the old journal, the Journal of Irreproducible Results?
I do. I certainly do.
I wish they'd bring that back. That was
Well, this
absolutely
is
spectacular.
the Mark Abrams runs the heir to that tradition, the Journal of Annals of Improbable Research.
Right.
Excellent. Okay, there are our picks. Go
then-
off and check them. Yes, Dick.
So if Elvin Cabot were still alive, unfortunately he's not, but he was one of my favorite gurus here at Columbia. His favorite journal was Acta Retracta.
Acta Retracta. That's great. Acta Retracta. I wish we could name this episode that, but
Please.
Acta Retract. No, there's nothing in this episode that should be retracted.
No.
If you'd like to find some other nice science podcasts, head on over to sciencepodcasters.org or promednetwork.com. We're members of both sites, and they have other good stuff there as well. Gentlemen, thank you so much.
Pleasure.
Thank you. Always a good time.
Alan is at dovedocs.com,
Yep.
where he blogs relatively frequently about what's on his mind, and you should go check him out there. Dick is in my office. He's also at verticalfarm.org.
Come.
.com, Trichinella.org,
Yep.
and medicalecology.org.
Correct.
You should check them all. His media empire, waiting for a bid from...
Rupert Murdoch.
Thank you. I just can't remember these things at all.
Unfortunately, I can.
We have a website, TWiV.tv, where you can find show notes and links.
Oh.
Dick, what's up?
We forgot to mention Radiolab. It was rebroadcast today.
Oh, your episode?
Yeah.
Hey, Alan, did you hear Dick on Radiolab?
No, I didn't.
He did an episode on parasites a few weeks ago.
Oh, I'll have to check that
it out.
out.
It's very
It was
good.
fun. I'm not the only one on there. You'll know the other guys too. And I'm blocking on his name, of course, and this is being recorded, so this is too bad.
It's all right. And I'm not cutting it out, It's too much work.
Of course it is. So
Don't
I'll
worry
just
about
have
it.
to suffer. I'll call you at 3 a.m. and let you know
It's
who it
really
was.
good episode. Check that out. We should get this guy, Dick Despommier, on TWiV.
You think?
Yeah, sometime. TWiV.tv. Send us your questions.
I hear
and
he's a bit of a
comments.
loose cannon, though.
Loose cannon.
Very much so.
I can't make up any of these double entendres.
I can't even make up a single entendre.
Well, you do cook well.
Thank you.
I
take
know.
that.
I had dinner at Dick's house last week. He's really a good cook. Excellent. Can I come back sometime?
Absolutely.
Thank you very much.
Absolutely.
We could do a TWiV from your house. Send us your
Let's do
comments
that.
and questions at TWiV.tv. Please go to iTunes and subscribe. It really helps us. And if you haven't done so and are able to, leave a comment on iTunes. It helps bring us to the front page of iTunes. You've been listening to This Week in Virology, all about viruses. Thanks for joining us. We will be back next week. Another TWiV is viral.